Good day, this is Professor Will, CBMD PhD. Today is Monday, July 27, 2020. It is 2:40 and 20 seconds a.m. Eastern Standard Time, and this is a continuation of my corona research. But before we get started, let's take a look at something I wrote. In today's video diary, I would like to express a much more deeper understanding of my passion for computer programming. In my deepest past, dating back to the mid-1980s, I had a passion for writing ASCII animations and batch files. Throughout my childhood years, there has always been sporadic gaps with my academic independent and ongoing learning. Today I feel there has been something missing in my life. I always wanted to go back to the mid-1980s, VIC-20 programming and study the foundation of mathematical algorithms that make computers possible. I had programmed computers sporadically over the last 35 years, but just over the last few days revisiting those ASCII algorithms and combinatorial mathematics. It has been real fascinating. And just over the past weekend, I managed to solve an algorithm to a problem that has puzzled me for quite some time. Past dating a few years back when I was taking up studies at the university on neuroinformatics and microbiology. The new program will be written under my name as my intellectual property. Well, I hope my audience enjoyed my presentation. Hope to see you again. Until next time, let's continue on with my corona research.
Lab-made virus mimics COVID-19 virus. Is safer to work with can aid efforts to find drugs, vaccines. Date July 21, 2020. Source Washington University School of Medicine. Summary. Researchers have created a virus in the lab that infects cells and interacts with antibodies, just like the COVID-19 virus, but lacks the ability to cause severe disease. This safer virus makes it possible for scientists who do not have access to high-level biosafety facilities to join the effort to find drugs or vaccines for COVID-19. Airborne and potentially deadly, the virus that causes COVID-19 can only be studied safely under high-level biosafety conditions. Scientists handling the infectious virus must wear full-body biohazard suits with pressurized respirators and work inside laboratories with multiple containment levels and specialized ventilation systems. While necessary to protect laboratory workers, these safety precautions slow down efforts to find drugs and vaccines for COVID-19 since many scientists lack access to the required biosafety facilities. To help remedy that, researchers at the Washington University School of Medicine in St. Louis have developed a hybrid virus that will enable more scientists to enter the fight against the pandemic. The researchers genetically modified a mild virus by swapping one of its genes for one from SARS-CoV-2, the virus that causes COVID-19. The resulting hybrid virus infects cells and is recognized by antibodies just like SARS-CoV-2, but can be handled under ordinary laboratory safety conditions. Researchers distributed the virus to researchers in Argentina, Brazil, Mexico, Canada, and of course all over the U.S. We have requests pending from the U.K. and Germany. Even before we published, people heard that we were working on this and started requesting the material. This virus is a workhorse of virology labs because it is fairly innocuous and easy to manipulate genetically. Primarily a virus of cattle, horses, and pigs. VSV occasionally infects people, causing a mild flu-like illness that lasts 3 to 5 days. Viruses have proteins on their surfaces that they use to latch onto and infect cells. The researchers removed VSV's surface protein gene and replaced it with the one from SARS-CoV-2 known as Spike. The switch created a new virus that targets cells like SARS-CoV-2, but lacks other genes needed to cause severe disease. They dubbed the hybrid virus VSV SARS-CoV-2, using serum from COVID-19 survivors and purified antibodies. The researchers showed that the hybrid virus was recognized by antibodies very much like a real SARS-CoV-2 virus that came from a COVID-19 patient. Antibodies or sera that prevented the hybrid virus from infecting cells also blocked the real SARS-CoV-2 virus from doing so. Antibodies or sera that failed to stop the hybrid virus also failed to deter the real SARS-CoV-2. In addition, a decoy molecule was equally effective at misdirecting both viruses and preventing them from infecting cells. Humans certainly develop antibodies against other SARS-CoV-2 proteins, but it is the antibodies against the spike that seems to be most important for protection. So as long as the virus has the spike protein, it looks to the human immune system like SARS-CoV-2 for all intents and purposes. The hybrid virus can help scientists evaluate a range of antibody-based preventives and treatments for COVID-19. The virus could be used to assess whether an experimental vaccine elicits neutralizing antibodies, to measure whether a COVID-19 survivor carries enough neutralizing antibodies to donate plasma to COVID-19 patients, or to identify antibodies with the potential to be developed into antiviral drugs. One of the problems in evaluating neutralizing antibodies is that a lot of these tests require BSL-3 facility in most clinical labs, and companies don't have BSL-3 facility. With this surrogate virus, you can take serum, plasma, and antibodies and do high-throughput analysis at BSL-2 levels, which every lab has, without a risk of getting infected. And we know that it correlates almost perfectly with the data we get from bona fide infectious SARS-CoV-2. Since the hybrid virus looks like SARS-CoV-2 to the immune system but does not cause severe disease, it is a potential vaccine candidate. Sputum testing provides higher rate of COVID-19 detection. Date July 24, 2020. Source: Brigham and Women's Hospital. Summary: in meta-analysis, researchers found that sputum was more accurate than neuropharyngeal or oropharyngeal swabs. The study also shows early testing increased rates of COVID-19 diagnosis. Early and accurate detection is critical for preventing the spread of COVID-19 and providing appropriate care for patients. Nasal pharyngeal NP swabs, which require inserting a long shaft into the nasal cavity to collect a sample from the back of the nose and throat, are currently the gold standard for collecting a specimen for diagnosis. But the procedure is technically challenging, often uncomfortable for patients, and requires personal protective equipment that may be in short supply. Other approaches to collecting specimens, including from an oropharyngeal swab and sputum, have been tested in small studies, but there is uncertainty about which method is best for detecting the virus. Researchers conducted a systematic review and meta-analysis analyzing data from more than 3,000 specimens to compare the three approaches. The team found that sputum testing detected the RNA of the virus that causes COVID-19 at significantly higher rates, while oropharyngeal swab testing had lower rates. Regardless of the collection method, the earlier samples were collected after symptoms began, the higher the detection rate. The accurate diagnosis of COVID-19 has implications for healthcare, return to work, infection control, and public health. Our gold standard in and out of the hospital is the neuropharyngeal swab, but there is a lot of confusion about which sampling modality is best and most sensitive. Our study shows that sputum testing resulted in significantly higher rates of SARS-CoV-2 detection and supports the use of this type of testing as a valuable method for the diagnosis and monitoring of COVID-19 patients. Researchers scoured the literature, both preprints and published papers, for studies that accessed at least 2 respiratory sampling sites, using an NP swab, oropharyngeal swab, or sputum. From more than 1,000 studies, they identified 11 that met the criteria. These studies included results from a total of 3,442 respiratory tract specimens. The team examined how often each collection method produced a positive result. For NP swabs, the rate was 54%, the oropharyngeal swabs 43%, for sputum 71%. The rate of viral detection was significantly higher in sputum than either oropharyngeal swabs or NP swabs. Detection rates were highest within 1 week of symptom onset for all 3 tests. When it comes to testing, the earlier the better, as diagnostic accuracy is improved earlier after symptom onset, regardless of the sampling site. Unlike antibody testing, it is very rare to have a false positive qPCR test when diagnosing COVID-19 early in the course of the disease using these methods. Nasal pharyngeal swabs are collected through the nasal cavity. Oral pharyngeal swabs are collected by inserting a shaft through the mouth. And sputum samples are generally collected by having a patient cough deeply to produce and expel phlegm. Not all patients are able to produce a sputum sample. For such patients, a nasal pharyngeal swab may be the best collection method. The meta-analysis included only studies conducted on hospital individuals. Additional study will be needed of patients who are asymptomatic or have mild symptoms. The current study did not assess alternative testing methods such as saliva or anterior nasal swabs taken from the front of the nose. The holy grail will be to find a test that is readily acceptable by patients, easy to collect, and highly sensitive. Homemade face masks likely need at least 2 layers to curb COVID-19 spread. Reduces dispersal of airborne and viral droplets from nose and mouth, with 3 layers preferable. Date July 23, 2020. Source: BMJ. Summary: homemade cloth face masks likely need a minimum of 2 layers, and preferably 3, to prevent the dispersal of viral droplets from the nose and mouth that are associated with the spread of COVID-19, a new study indicates. Viral droplets are generated during coughing, sneezing, or speaking, and face masks are thought to protect healthy people from inhaling infectious droplets as well as reducing the spread from those who are already infected. But worldwide shortages of personal protective equipment during the pandemic have led some health agencies, such as the U.S. Centers for Disease Control (CDC), to recommend the use of homemade cloth face coverings as an alternative to surgical face masks. Several types of material have been suggested for these, but based on little or no evidence of how well they work. The researchers used a tailored LED lighting system and high-speed camera to film the dispersal of airborne droplets produced by a healthy person with no respiratory infection during speaking, coughing, and sneezing while wearing each type of mask. The video recording showed that the 3-ply surgical mask was the most effective at reducing airborne droplet dispersal, although even a single-layer cloth face covering reduced the droplet spread from speaking. But a double-layer covering was better than a single layer, reducing the droplet spread from coughing and sneezing, the recording showed. Nevertheless, based on their observations, a homemade cloth mask with at least 2 layers is preferable to a single-layer mask. They say, guidelines on homemade cloth masks should stipulate multiple layers. And the researchers emphasize there is a need for more evidence to inform safer cloth mask design, and countries should ensure adequate manufacturing or procurement of surgical masks.
Good day, this is Professor Will, CBMD PhD, on microbiology, infectious disease, and computer science. Well, hope you enjoyed listening. Until next time, have a great day.